All right, thanks so much, Janice. So as Janice mentioned, today we're going to be talking about an overview of the NovaSeq 6000 system. So before we dive in, I do want to mention that this is a pretty high-level overview of the NovaSeq. So if you've been running the instrument for a while, for example, I would definitely recommend joining our NovaSeq 6000 Run Quality and Best Practices webinar that should be taking place in a couple weeks. There's a lot of really great troubleshooting in that webinar. So for today, for this webinar, we will be going over a system overview, what reagents are available for the NovaSeq system, the NovaSeq workflow. So this will be ranging from upstream library, QC, and loading, as well as briefly covering some available analysis options. And then finally, we'll lightly touch on some maintenance and best practices for this instrument. So on to the NovaSeq 6000 system. So the NovaSeq uses pattern flow cells, which were introduced on the HiSeq X and are now used on the HiSeq 4000, iSeq 100, and NextSeq 2000 systems. The instrument utilizes two-channel SBS chemistry, which will be familiar to those of you that have a NextSeq platform, and it's also utilized on the MiniSeq as well. The NovaSeq uses cartridge-based consumables, which were first introduced with the MiSeq platform. And much like all of our other platforms, the NovaSeq has a touchscreen for ease of use when navigating through the software graphical user interface. For very high throughput labs, the system is also ready to integrate with a LIMS system, whether it's Clarity LIMS from Illumina or one developed from your lab. Finally, to help ensure the uptime of this instrument remains as high as possible, we have modularized hardware components to optimize the serviceability of the instrument. It's also capable of connecting with Illumina Proactive, which helps us monitor the instrument's hardware performance and will send alerts to tech support if anything looks to be operating outside of our hardware specifications. This in turn will help us potentially catch any issues that may arise, and we'll be able to resolve it for you before it affects the sequencing run. So now we're going to take a look from top to bottom of the instrument hardware. So from the top, we have our touchscreen monitor, easy to read and interact with. Directly beneath this is the instrument's optical bench. So this houses all of the hardware in charge of exciting the fluorophores and capturing the signal they emit in the form of images. Moving our way down from the optics is the dual activated deck or flow cell stage, where the flow cells are loaded for sequencing. The NovaSeq is able to run 2 flow cells at a time, exactly like our HiSeq platforms. The flow cell stage fully automates the flow cell loading process, such that all you will need to do is place the flow cell on the deck. The deck will automatically clamp the flow cell securely in place, as well as applying vacuum and biases it to the exact position for imaging. The remaining parts of the instrument houses all of the fluidics infrastructure, and each side has its own fluidics circuit. We have compartments for the cluster and paired-in reagent cartridges, which are housed in the reagent chiller. And just as a note, the reagent chiller is only on during the sequencing run. Beneath these cartridges is the buffer cartridge, which contains all buffers needed for sequencing, as well as for the automatic post-run wash. Apart from the buffer cartridge, we also have 2 different containers to catch the spent reagents. The larger container houses all of the non-hazardous materials, while the smaller one houses the formamide. We'll discuss these in a little bit more detail in the maintenance section. We have also built in a second method of spent reagent collection for high throughput labs, where the back of the instrument has connector points where it can be hooked up directly to a large waste drum. Now, coming back to some of the key features of the NovaSeq instrument, as I mentioned, we're utilizing two-dye chemistry. And some advantages of two-color chemistry are that it reduces the amount of images needed to be captured during sequencing, and thus this reduces runtime. It also helps to reduce the data footprint of the sequencing runs. So this system also uses all cartridge-based consumables. We'll talk in depth about what is included in each kit, but I did just kind of want to mention. Each consumable is RFID tracked so they can easily identify themselves to the control software during the run setup. Reagent information is automatically stored in the run parameters of each run for traceability. And these files will contain the reagent serial number, the lot number, and expiration date. The NovaSeq also utilizes pattern flow cells, which help with more data output and shorten the runtime and optimize base calling. The nanowells are both top and bottom surfaces of the flow cell glass, such that clusters form on both surfaces. Each well houses one cluster or one read, and there is no template generation needed since the NovaSeq has the map of the nanowells preloaded in the software. Rigid registration is performed to compare which wells are lit, indicating that it has a cluster, and which wells are not lit, indicating no cluster. This also gives us the ability to know the occupancy of the flow cell. For more information on pattern flow cell technology, please see the live link at the bottom right corner of this video, which has a really nice video presentation on pattern flow cells. So currently, we have a few different configurations of NovaSeq flow cells that are all in a 2-lane or a 4-lane format. If utilizing a standard workflow, we're loading the same pool in all of the lanes of each flow cell. If this isn't the best fit, we also have an XP workflow option where different sample pools can be loaded into each lane of the flow cell. And more to come on this just in a few minutes. So this slide has some specifications, but it also will help us look at the different NovaSeq flow cell configurations in a little bit more detail. The SP, S1, and S2 flow cells, they each have 2 lanes, while the S4 flow cell has 4 lanes. Now, if you notice from the image, the S2 flow cell lanes are wider compared to the SP and S1, but thus it has a higher data output capacity. Along those same lines, the S4 flow cell not only has more lanes, but each lane is wider compared to the SP, S1, and S2. So this gives the S4 flow cells the highest data output at 3,000 gigabases or 3 terabases of output. This is equivalent to 25 human genomes at 30x coverage. You'll notice that all of the flow cells have plastic casings around the edges, which allows the instrument to effectively grab the flow cell and engage. So while this platform is able to generate the most data of any of our instruments, the quality is still very much comparable to our other Illumina platforms that you've come to expect. We also have a full list of our NovaSeq 6000 specifications linked at the bottom here. So here I just kind of wanted to call out that some application-specific suggestions for SP, S1, and S4 flow cells. The SP and S1 flow cells, they do have a lower output range for smaller projects or pilot studies with a fairly fast turnaround time. So once the proof of concept of the pilot study has been established, the application can be scaled up for production runs with the S2 or S4 kits. For those of you that might be moving from the HiSeq 2500, here's a comparison of the HiSeq 2500 rapid run mode and how that can translate to the NovaSeq. So the SP kit has an output from 65 to 400 gigabases, depending on the read length. This is currently the only kit capable of 500 cycle runs. At the lower output range, the instrument is also capable of smaller projects or pilot studies with the faster turnaround time. So one of the main takeaways from this slide, if we were just to compare the output from the rapid run to that of the SP flow cells, we're able to achieve the same level of data quality at almost half the time and double the amount of data output. So the last thing I wanted to highlight about the NovaSeq system are the two sequencing workflows. So we have the standard workflow and the XP workflow. The standard workflow has a streamlined operation with less hands-on time and automated XAMP mixing. So this is because with the standard clustering workflow, you're loading the same pool in all of the lanes. So the standard workflow has the same library across all of the lanes because of the fluidics infrastructure on the instrument. Now, in order to provide more flexibility, we also offer the XP workflow. So this allows you to sequence different library pools in different lanes. So as discussed earlier, this can be useful when you don't need a high amount of coverage per sample. Another advantage of the XP kit is that it requires less sample input than the standard workflow. So the XP workflow is very similar to the CBOT Duo for those of you familiar with the HiSeq 2500. In order to perform the XP workflow, you won't need a CBOT, but you will need to make a one-time purchase of a flow cell loading dock. You'll also need to purchase one XP loading kit per flow cell. And these kits will come with the flow cell manifold and a set of XAMP reagents. With the XP workflow, you will manually set up the XAMP. When the library pools are mixed with the XAMP reagents, they're manually loaded onto the flow cell lanes via the dock and the manifold. Once the samples are loaded, the flow cells should be moved to the NovaSeq and the run started within 30 minutes, as the XAMP is fairly time sensitive. The XAMP reaction is very efficient, and unregulated it can cause a decrease in quality for the run. So some of you may have seen the release about the new NovaSeq 1.5 reagents that are coupled with our new control software version 1.7. These were released a couple of weeks ago. The V1.5 reagents do require the new control software version 1.7, but this new control software version is compatible with both the current V1 reagents and the new V1.5. So some of the highlights of the new kits are that we've increased the number of cycles of SBS. So currently our kits have an overfill of 30 cycles for indexing and dark cycles. But the new 1.5 reagents will have an overfill of 38 cycles of SBS. So for example, your 100 kit 100 cycle kit will have 138 cycles of reagents. We've also released a new version of Illumina Experiment Manager for creating sample sheets. This will help with some of the indexing changes that we'll talk about on the next slide. Next, we've introduced a S4 35 cycle kit. When you combine that with the extra 38 cycles of SBS, they will have a total of 73 cycles. We've also updated the Q30 specifications. So for most of the kits, like the 2x100 and 2x150, the Q30 has increased to greater than 85%. Previously, it was 80%, I believe, for both of these kits. So we've increased these specifications to be more in line with what customers are seeing in the field. Next, to touch on the new software version, this software will be user installable. The release notes, the update instructions, and the installer can be found at this link here. If your lab is able to do desktop or screen shares, tech support is always happy to help with these upgrades as well. So one of the major changes with the new reagents and software are the updated indexing strategy. So currently, our V1 reagents follow the forward strand workflow like we have on the MiSeq and HiSeq 2500 system. This uses a grafted oligo for the I5 index. With the 1.5 reagents and the 1.7 software, we're changing to the reverse complement workflow like we have on the NextSeq, MiniSeq, iSeq platforms. Down below, I've included a quick reference chart just to kind of keep all of the different versions together. It's a lot to remember, but I just wanted to highlight that on 1.7, if we're still running the V1 reagents, we're going to continue to use the grafted oligo indexing workflow, which includes the need for 7 dark cycles. So we'll have to take into account those dark cycles when figuring out our total number of cycles of SBS chemistry. Now, when switching to the 1.5 reagents, we no longer need the dark cycles for our Index 5 primer. This will also allow us to use a custom primer for our I5, which we are unable to do with the current V1 reagents. Okay, so now that we've kind of gone through an overview of the instrument and some of the kind of exciting new features, let's talk about the sequencing kits available and what those include. So again, the NovaSeq leverages all of the improvements that we've made on previous sequencers for ease of use for the user. So we're really bringing the high output instruments to a lower touch point in terms of high efficiency. So to perform one run, we'll need to purchase one NovaSeq kit. Currently, we have the following types of sequencing kits based on various flavors of flow cells. This chart also notes the number of SBS cycles available with the V1 and V1.5 reagents. So as I mentioned earlier, the SP kit is our kit with the longest run configuration. So it has the longest supported run length of 2x250 for a total of 500 plus sequencing. Our S1 and S2 each come in a highest supported read length of 150, and these come in 100, 200, and 300 cycles. And then our S4, which was our highest output reagent kit, comes in the 200 and 300, and then the newly released 35 cycle kit. And again, I just wanted to mention that for the V1 reagents, we do have to account for the 7 dark cycles if we're doing dual indexing. So each NovaSeq kit comes with 5 items. So you'll have your cluster and paired-in reagent cartridge, your SBS cartridge, your flow cell, library tube, and buffer cartridge. The cluster and SBS cartridges are stored at -20 until they're ready to be used. They're roughly the size of a MySeq cartridge or a standard shoebox. The flow cell is stored at 4C, and it's about the size of about 1.5 times the size of a NextSeq flow cell. The library tube and buffer cartridge are stored at room temperature. And note that the buffer cartridge is about the size of a picnic basket and can weigh up to 10 pounds depending on the kit, just due to the sheer amount of volumes needed to accommodate the large flow cell size. If you're a part of a very high throughput lab, or you just need to have a constant stock, all of the box dimensions for the kits, they're listed in the site prep guide, and these can help with storage space planning. So here's a little bit more detailed look at the cluster cartridge. So these cartridges are keyed in such a way that they can only be loaded into the instrument in one orientation. Every other row of wells are empty, and this is for the automatic post-run wash, where buffer is being pushed through the lines and into the empty wells. Reagents are used from back to front throughout clustering and paired-in chemistry. The library tube will be loaded into position 8, and positions 5 through 7 are reserved for custom primers if needed. Position 17 contains the sodium hypochlorite or bleach, and this is used for the automatic post-run wash. So it is diluted to 0.12% with buffer before being used to make sure we clean out the template line to prevent sample carryover. We also have a removable position, position 30, for the formamide, similar to the NextSeq and MiniSeq cartridges. The SBS cartridge contains the incorporation mix, scan mix, and cleavage mix in the listed positions. And just like the cluster cartridge, it also contains an extra well for each reagent to catch the wash buffer during the automatic post-run wash. Also, just like the cluster cartridge, it is keyed in such a way that can only be loaded in one orientation. For the NovaSeq, another cool new feature is that the scan mix is now degassed on the instrument to help prevent bubbles during imaging. So the buffer cartridges are a picnic basket that we talked about earlier. And just like the cluster and SBS cartridges, it can only be loaded in one orientation. So this is from where we draw the wash solution for the automatic post-run wash. And it also contains the buffers used during the run itself to remove reagents from the previous chemistry step. Here's a detailed view of the XP workflow kit options. So we have a 2-lane and a 4-lane XP loading to match the flow cells. So the 2-lane XP kit can be used for SP, S1, and S2 flow cells, while the S4 flow cells will need the 4-lane kit. So as previously mentioned, you will need a loading dock, which that's the one-time purchase dock, and it can be used anytime we need to make flow cells via the XP method. We also sell 2- and 4-lane extra manifolds, just in case we need some of those on hand. All right, now that we've learned about the instrument and reagents from an overview, let's take a look at just kind of an end-to-end workflow. So the NovaSeq is an open platform, which means that we don't limit the type of application for this system. All Illumina libraries are compatible, and theoretically, any library containing the Illumina P5 and P7 adapters are compatible, so optimization will likely be needed. If we're working with customer third-party libraries, it'll be up to the user to validate this library type on the NovaSeq. So just a few library considerations. The first consideration is loading concentration. If we're running a 100% Phi-X run, we use a 250 picomolar loading concentration going into onboard cluster generation. As with all Illumina platforms, shorter libraries have higher clustering efficiency. So depending on the size, the loading amount should be adjusted accordingly. When sequencing low diversity libraries, best practice is to start with 10 to 20% Phi-X or another well-balanced library and further optimize from there. If we're doing methylation sequencing, 10% Phi-X should be sufficient. So similar to other platforms, if we're using low-plex pooling, we do recommend that bases from both red and green channels are represented. If the libraries have already been diluted and denatured and transferred into the library tube, but the run was not able to be started as planned, that library tube can be popped out of the cluster cartridge and saved at either -20 or 4C for a later time. For optimal results, we should use the sample as soon as possible, though. Good library QC is crucial for successful sequencing results. And as with all of our other Illumina platforms, we do recommend to use a fluorometric method for quantification to detect double-stranded DNA. For even more accurate measurements, consider qPCR, as this will only measure full-length libraries which are clusterable. We also recommend to QC the quality of the library on a bioanalyzer to confirm the library size and check for unexpected contaminants. We don't recommend to use a UV spec for QC, as this tends to not be specific enough for sequencing. If you have any questions about your specific library type, we would recommend to reach out to our library prep support team, and they can always discuss the best method for quantification of that type. So before pooling our libraries, we should first consider how much library we want to load onto the flow cell. Just a little bit backwards compared to previous platforms, but if you're familiar with the HiSeq 4000 or X, it'll be similar to that with those pattern flow cell systems. But the library input amount for these flow cells are comparatively higher due to the sheer size of the NovaSeq flow cells. We don't want to run out of sample after pooling. In this chart, we first have the desired final loading concentration to the flow cell in picomolars on the left, and the corresponding starting concentration of that pool. So for example, if I want to load my samples at 300 picomolar for onboard cluster generation, I will need to create a pool at 1.5 nanomolar. Once I've made my pool of samples at 1.5 nanomolar, this will be taken for denaturing with NaOH and neutralization with tris. The final loading volume going into the library tube varies for flow cells, so it will vary by flow cell type, and This flow cell will then go into the supplied library tube and then into the cluster cartridge as well. Illumina also has a pooling calculator to help with pooling of each library if needed. You can simply enter the plexity desired for the pool and whether the concentrations of the individual samples going into the pool have been normalized or are different. Then we will enter the target concentration and total volume of the pool. This calculator will help provide how much of each sample to add for the pool based on its starting concentrations. And please note that this is not particular for the NovaSeq platform, so we can really use this with any of our instruments. So after the library has been denatured and diluted, we're now going to prepare for the actual sequencing run. So first we will need to thaw our cluster and SBS cartridges. These can take a while, so we may want to take them out to thaw before we denature and dilute our libraries. The cartridges do tend to float in a water bath, so using wire racks can prevent them from tipping over in the water bath. Once they're thawed, confirm that there's no residual ice chunks, and it's really important to mix all of the reagents that full 10 times to make sure they're homogeneous, as this can affect the read-1 sequencing quality if they're undermixed. Since the buffer is stored at room temperature, it can be taken directly from storage and loaded onto the instrument. If custom primers are needed, they should be loaded into the reserved wells or spiked into the Illumina primers. In the resources section at the end, we do have the custom primers guide, and it's also linked here. And we also have a bulletin for spiking those custom primers into the Illumina primer wells. And I do also just want to highlight again that with the new 1.5 reagents, we're now able to use a custom primer for the I5 index, which was previously unavailable in the V1 reagents. So next we'll remove our flow cell from 4 degree and let it sit at room temperature for 30 minutes. This helps to prevent condensation on the glass during the run. We'll remove the flow cell from its plastic housing and clamshell, and it's very important to inspect the flow cell on both the front and back for any potential debris. So if any debris, such as small glass shards, for example, are seen, we can clean the glass surface and gasket areas with lint-free tissue paper. If the run cannot start immediately after the flow cell has been opened, it can be put back at 4 degree and used within the next 24 hours. Next, we'll load the denatured and diluted library pool into the library tube. And we will be sure to leave the cap off. We'll load that into the cluster cartridge. And one nice thing is that if we did forget and put the cap back on, the cartridge will not fit into the instrument. So once all of the consumables are ready, we'll load them onto the instrument as prompted. So it will take a little while for the RFID information to register during run setup. So now that the run is ready to be set up on the NovaSeq instrument, let's just take a brief tour of what the control software looks like. So to the top left will be the hamburger menu. And from this menu, we can minimize the software to access Windows. We can close the software, which will exit to Windows. We can initiate a power cycle. And we can also check the data transfer status as well as change some settings. Under the Settings menu, we can toggle between a few options for data analysis and storage. The first option is Manual mode. So in this mode, the base call files and sequencing data will be sent directly to a network location. Due to the extremely high amount of data generated from this instrument, we can't set the output location to be on board the local system. It has to be offloaded to a network or to base-based sequence hub. The second option is file-based mode. So if your lab has a LIMS system, whether it's Clarity, LIMS, or self-developed, this is where it can be integrated with the instrument. Base-based Sequence Hub is available for both manual and file-based mode in the Run Monitoring Only or the Storage and Analysis options. Stepping out of the Settings menu and going into Process Management, this is where we can monitor the run progress as well as data transfer progress to base-based or the network. Before starting any new runs, it's best practice to come here and verify that all processes are complete. The NovaSeq utilizes a Linux-based server called the Compute Engine to store sequencing data locally prior to transferring. Because of this, the only method to curate the CE is to use process management and to delete completed runs. The CE has a storage capacity of 2.9 terabytes, which can store up to 2 full S4 300 cycle runs if for any reason data transfer is interrupted. Because of this, we do want to make sure that the drive space is monitored closely and that we not start any further runs until the prior runs have been offloaded. RTA also lives on the CE, and this is the software that performs our base calling, which converts the raw images into base call information. NovaSeq uses RTA3, which makes concatenated base call files, which are 1 per lane per cycle. And this helps to reduce the data footprint. So for more on how this works, we have a white paper listed at the end that goes into a little more detail of RTA3. So now that we've kind of taken a little bit of a tour of the hamburger menu, let's walk through the run setup. As you can see, the screen itself is very self-explanatory and intuitive. So depending on how many runs you want to set up, we can select which side we want to load. So either A or B, or we can set up both A and B at the same time. At this point, we can just follow the screen prompts. There's also some videos that go along with those. One thing I do want to note at this step is just to make sure that there's nothing sitting on the slab, which is the gray part of the instrument, because this can potentially interfere with the flow cell door operations since it's motorized. So we'll load the consumables as prompted. And again, that RFID reading, it tends to not be instantaneous, so it's normal to have a slight delay as things are being loaded. There will also be a reminder to empty the spent reagent containers before we start a new run. This has to be acknowledged in order to proceed. And if any of the containers become full during a run, the run will abort to prevent them from overflowing. So it's really important to make sure that they're empty. So once the RFIDs have been read, they will be displayed on the screen, and we can now proceed to the next step. So next we'll be given an option to use base for these runs by entering your account credentials. As I mentioned earlier, you can use base for run monitoring only or both run monitoring and storage. So next we'll be defining the run parameters, such as the run name, the number of cycles, the network output location, and sample sheet. The sample sheet is optional unless the run is being sent to BaseSpace. So if we're using the cloud for analysis, then a sample sheet will be required. For the output location, we can no longer use a map network drive. So what we would recommend is to use the UNC, or the Universal Naming Convention path, for your desired network location. Also, the software will prevent us from using the C, D, or Z as output locations because these drives are local to the instrument. So once all of the parameters have been inputted, we'll be given an option to review all of the information to make sure they're correct. Really important to note here that once we select Start Run, we'll no longer be able to edit any of this information or go back. After the Start Run is selected, the instrument will first go through a series of pre-run checks to make sure that all of the hardware, network connections, drive space are good to go. When all the tests pass, the run will automatically start. And if any particular check fails, you'll be given the option to retry that test. This is what we see when the run actually begins. So on top, we have our estimated completion date and time, in case we're planning ahead for our next run. We also have some high-level metrics, such as the cluster's passing filter, the overall yield, as well as intensity and Q30 by cycle once the run gets sufficiently far. At this point, we can walk away and we don't need to return until we're ready to set up the next run. So here's just a quick overview of the estimates for various steps. So the clustering takes about 3 hours. The SBS is anywhere from about 4 to 6 minutes per cycle, depending on the flow cell type. Chemistry is now our rate limiting step just due to the improved optical hardware. So this helps us to speed up the imaging time. Curtain turnaround is about 40 minutes as well. So once the sequencing run is complete, if the data was streamed to BaseSpace, we have the option of generating FASTQs in the cloud and then taking it to various analysis apps. Or for a more streamlined workflow, Clarity LIMS will also automate the downstream processing. If you have a custom-built pipeline, the base call files on the network can be converted to FASTQs via BCL to FASTQ, for example, and port it over to your analysis pipeline for further processing. Now we're just going to touch on the post-run procedures. So once a sequencing run is complete, a post-run wash is automatically performed by the instrument. The instrument wash utilizes solutions in the buffer cartridge as well as the sodium hypochlorite from the cluster cartridge, and the overall wash takes about 80 minutes. So as a refresher, the spent reagents are separated by non-hazardous and spent formamide reagents via the large and small spent reagent containers. We do highly recommend that these are emptied between each run. The small spent formamide reservoir contains about 90 milliliters of waste per run. So of this volume, about 10% is formamide, and the other 90% is wash buffer used to clear the instrument fluidics. The drawer pulls out for access to the large non-hazardous spent reagents. The formamide reservoir can be removed by lifting the lever on top of the bottle. We also have that built-in detachable formamide position on the cluster cartridge that we talked about earlier. So this is position 30, And to remove it, you'll simply lift the white plastic to detach and the clear plastic to release. So finally, we'll just touch over some maintenance and best practices. So we'll talk about washing and idling the system here. So we already discussed that the instrument will automatically perform a post-run wash, but in addition, we should be performing a maintenance wash if one of the following circumstances are met. So if the instrument has not had a run per side for more than 14 days, if the instrument has not had a 4-lane run per side in the last 14 days. So if your lab is consistently running, say, S1 or S2 flow cells, we will still have to perform this maintenance wash every 14 days to wash all 4 lanes. The third case in which we'll have to perform a manual wash is if a run ended prematurely. So when performing a manual maintenance wash, please be aware that the system does need a used buffer cartridge. So we do recommend that labs save a couple of used buffer cartridges from your old runs, just in case we need to perform some extra maintenance washes. Here's just the overview of preparing that wash solution and how to get that ready. And then once the tween and sodium hypochlorite are made, we'll load those cluster and SBS wash cartridges and that used buffer cartridge from the run. We'll also be using our 4-lane wash flow cells to set these up. Now, typically when the instrument is not in use, we do recommend to just leave it on. In this state, there are temperature controls that are maintained. But if we do need to shut down the instrument for any reason, such as a planned power outage, or if we need to do a routine power cycle through troubleshooting, we can follow these steps to perform that power cycle. So we'll shut down the instrument through the control software under the Shutdown Instrument tab. Once the computer is turned off, we will use the toggle switch at the bottom right of the instrument. We'll leave that off for a minute. And in the case of a power cycle, after that minute, we'll turn everything back on in reverse. So we'll use the toggle switch to boot up the instrument, and then we'll use the power button on the right side to boot up the computer. So here's just a collection of the information from the last few slides, just in one kind of easy to look at place. Just to leave you with a few last thoughts, if you guys encounter any issues, particularly any RFID errors during setup, definitely give us a call. And we always recommend to use freshly prepared sodium hydroxide, and to ensure that the pH of that sodium hydroxide is above 12.5. We recommend to pre-qualify libraries using the iSeq, so it's just a really good QC method. And then load the libraries quickly after denaturation to avoid secondary structures. So finally, just some resources. So at the beginning, we linked the new 1.5 Q30 specifications, but I did want to leave you with the current specifications for V1 reagents. We also have our general resources. Here are some base-based public data sets. So I wanted to highlight these because we do have a whole list of new 1.5 reagent data. If your lab is going to be transitioning to those reagents soon, you can kind of see what the data quality and output looks like there. Here's some of our support bulletins. These are developed by tech support anytime we find a topic that might be really useful. And so some that I wanted to call out are what should I do if the flow cell cannot be loaded during run setup, how to set the output location, and plotting the percent occupied and percent pass filter to check your voting concentration for the NovaSeq platform. Lastly, thank you so much for joining. I did want to highlight again the NovaSeq 6000 Run Quality and Best Practices webinar. This is a pretty new webinar, but we got some great feedback last time, and it just has tons of troubleshooting advice in there. So if you have time, please check that out. And then at this point, we will take any questions.